Sí, bueno, para la tercera charla vamos a invitar al doctor Guido Junge. El doctor es cirujano en trasplante formado en Alemania. En realidad trabajó muchos años en el hospital de Charité en Berlín, en cirugía de trasplante renal y hepática, pero desde hace ya varios años es el jefe de medicina transnacional y autoinmunidad, tanto trasplante renal como para riñón, tanto para riñón como para trasplante, y trabaja para Novartis, Institute of Biomedical Research, y él nos va entonces a ilustrar sobre los targets clínicos y moleculares en enfermedades renales. Doctor Junge.
Let me start with a thank you for inviting us here, and first of all to the chairperson, and I would also like to thank the president and also the organizers for having us here. And what I also have to disclose, that none of the products of the target that we are discussing today are yet approved. So we are speaking literally about off-label use. I just wanted to record this at the beginning. I thought I'd bring you three new molecules or three new targets. And the first one is CD40, you have heard about this. Another one is an siRNA that is directed against p53, so basically trying to inhibit apoptosis. And then the third one is the complement system and targeting the alternative pathway in various renal diseases. And I brought a fourth topic, and we'll see how time works and whether we could also speak maybe about organ availability. And the first case is about anti-CD40, and I think we just heard this in the two prior talks. The immunosuppression that we're using today is pretty much targeted on T cells. We usually wipe or don't wipe out the T cells at the beginning, and then what we usually try, we have a combination therapy trying to control the T cell activation, and then we have a combination therapy to control also T cell expansion, either with an IMPDH inhibitor or we're using a TORC inhibitor. But we are kind of neglecting the T cell B cell interaction, and what we heard, especially in long-term and chronic rejection, we are not really controlling B cells and donor-specific antibodies. And what we have learned, and we saw this, short-term results, irrespective of whether we look on kidney on the left or liver on the right side, are excellent. 1, 3, and 5-year results are really outstanding good. But the problem that we are facing, and Dr. Maas was alluding to this, is that long-term outcome, even if we are looking at large registries and we see a slight improvement, are not really good. What we see is 30% of graft failure after 5 years in kidney transplant, and that increases up to more than 50% after 10 years. And also, if we look into the other population, if we look into liver transplantation, what we see is that after 5 years, 20% have chronic kidney disease. And this is interesting because they really have their native kidneys, and the renal toxicity that we're observing in liver transplant recipients is mainly an on-target toxicity of the immunosuppression that we are using. And this is obviously something where we say, could we do better? We could ask the question, can we do better? And I think the first approaches that we have seen was Bristol Myers Squibb, looking at co-stimulatory blockage and see, would we have something that would allow us to walk away from CNI immunosuppression and have a CNI-free regimen to may generate longer or better long-term outcomes and have less nephrotoxicity. And I think there is a debate, which target should we really target? Should we target CD28 or should we target CD40? And I think it has to do with safety findings, has to do with viral reactivation, and has to do with the virus-specific T cell response when you're blocking the CD28 pathway. And I think I just wanted to very quickly speak about hyperacute rejection and what we are seeing in the field at the moment. And I think what we learned with all the modern technologies, the typing technologies, and also the characterization of preformed antibodies is that we have identified a series of epitopes where normal people or people here in the room and people on the waiting list have preformed antibodies. And the question is, what would happen if we have a way to desensitize or create donor organs that are lacking the epitopes that we are finding. And I think the technology that people are using very effectively at the moment is CRISPR-Cas9, where you have very precise method to either knock out or insert genes. And what we are observing at the moment, and this is interesting, is a production of knockout pigments. And it's basically a cloning therapy, so you take a fibroblast line, you knock out the genes where you think you have preformed antibodies, you do a somatic nuclear cell transfer, and then you have an in vitro fertilization and have piglets that are lacking of the gene that you knocked out or that you knocked in. And there are interesting publications, and they were looking at the behavior of preformed antibody with the certain knockouts. And what we are seeing here is the preformed antibodies in patient on the waiting list, and then what would happen if you do knockouts. And what we see is if you do a single, a double, a triple knockout, you all of a sudden come into a situation that almost 80% of patients on the waiting list wouldn't have preformed antibodies. Whether this translates into really pig-to-human xenotransplant, I don't know, but at least we have technology that allow us to come closer to the situation. And I think also you just have seen this in the recent years. There are companies that really have focused on the knockouts of the PERF viruses. Yeah, exactly. And eGenesis published a fantastic paper. I mean, that's like Olympia. They knocked out all the 63 PERFs. This is really impressive. And we don't know where we are going, but I think we will see more and more of those approaches. And I know that the FDA was recently exploring a few of those proposals. And I just want to close this chapter before I'm going into really something that's interesting for us. Tector et al., they published last year their first series with pig into monkey transplant with a survival in kidneys of more than 400 days. Small series, but obviously encouraging. The one I think that is more interesting for us at the moment is complement system. And I think we have a few renal diseases where we know that complement plays a role, either essential or a bystander role. And for example, C3G glomerulopathy or DDD are most likely poster child for complement-driven diseases. 3 ways of activating complement system, the classic, the lectin, and the alternative pathway. And at the moment, only one treatment available that downstream blocks with an anti-C5 antibody the formation of the membrane attack complex. So there are other considerations when asking, could we target the immune system, the complement system at a higher level upstream of C5 and upstream of the membrane attack complex? And one of the promising targets is obviously factor B. Factor B inhibitor would inhibit the formation of the caspase, of the convertase that activates and assembles the downstream activities. And one of those compounds that is at the moment in clinical testing is called an LNP023, and this is a low molecular weight factor B inhibitor. And this is interesting. At the moment, the complement system is only attacked with injectables, so with a biologic, and this is actually an oral compound that effectively, and you see the IC50, that's 0.009, so it's a highly selective and effective human factor B inhibitor that is in clinical testing. And the first thing that you always do, and I just wanted to share a few data with you, is single ascending dose to really see how does a compound behave in humans, and then you do a multiple dose to see is that something that gives you enough exposure over time. And what you are seeing here with increasing doses, the compound behaves very well in a single dose. There is absolute dose and exposure correlation, and the same thing here in a multiple dosing. The other question that you always ask if you go with a new molecule into humans, how would the drug behave in a situation of fasted or non-fasted patient? Is there any food effect? Do you need to be careful with food effect? And those two curves completely overlapping, thus showing you there is no food effect for this oral factor B inhibitor. And then the other question is, and just flipping through, is the question, would you see any safety findings? And if you compare placebo and patients treated up to the highest dose, there's absolutely no discrepancy between the two groups. And then a last question that we are always asking before we're going into patient population, specifically into potentially renally impaired patient population, is the question, what is the metabolism, the breakdown, and the excretion of the compound? And if we are looking at this specific compound here, LNP023, what we are seeing is that the main metabolism, more than 80%, is really a hepatic breakdown, and the contribution for extrahepatic and renal excretion is very low, the 3 to 5%. So the risk that you have to adjust for impaired renal function is very low. And what I just wanted to show is what we call a proof of concept. Usually if you have a disease that targets a specific pathway, you usually look for a disease that's mainly driven by this pathway. And I think everyone would agree that a typical complement disease is paroxysmal nocturnal hematuria. And what we see in paroxysmal hematuria, it's an acquired somatic mutation that in the end leads to opsonization. P3B accumulates on the surface of red blood cells, and then you have the activation of the cascade, you form the membrane attack complex, and in the end we have hemolysis. Not only hemolysis, we also see thromboembolic events, and overall patients have a reduced life expectancy. The only treatment available at the moment we were speaking about this is eculizumab, that targets actually downstream at the C5, but it doesn't inhibit the opsonization of the red blood cells. So what we are seeing is we have a certain control, about 40% of all patients, because they are still opsonizated, we have still hemolysis, extravascular hemolysis in the spleen and in the liver. So this is a population that was targeted with the new compound, with LNP023. And if you just look at the analysis here, you see hemolysis increasing from top to down, and you see the C3 opsonization from the left to the right. And what we see in controls, we have massive hemolysis and we have massive opsonization. What we see with eculizumab, to a certain degree we control the hemolysis, but we still have the opsonization. We're not controlling opsonization. And if we look into the test compound LNP023, we have similar control of hemolysis, but we don't have any opsonization. And you see this sweet spot here where you would expect healthy red blood cells. The question is, how would this translate into humans? And this is just a simple proof of concept trial where you have a cohort of patients with persistent PNH despite treatment with an anti-C5 antibody. And what we are seeing here is various biomarkers that would show the target engagement, CH50 as a marker for the classical pathway, or VSLAP and BB and the C3 fragment as markers of the alternative pathway. And what we see in the 3 patient population, again hard to see, but you have the CH50 on top, and you have the VSLAP and the fragments in the middle and in the lower part. What you see immediately after treatment, basically within hours, you control the alternative pathway, you control the complement system, and all the values normalize. And the other question is, how would this translate into clinic? And I think one of the easiest marker for hemolysis is LDH. And what you see immediately after treatment, significant reduction in LDH. If you have single patient graphs, your spaghetti plots, you see, irrespective of the activity of the hemolysis, all these patients are controlled within 2 weeks. And this is also other parameters. If you look at haptoglobin, reticulocytes, bilirubin, all these parameters normalize within days. And this was for us the question. Obviously, the compound has target engagement. It does what it is expected to do. And then the question was, what would be the first kidney disease to attack? And we have selected 3 diseases. One is complement glomerulopathy, IgA nephropathy, and membranous nephropathy. And I just wanted to give you a short glimpse on how these trials are conducted. They are underway at the moment. It's a kind of a combination of a phase 2A/B. So it's not only a proof of concept with a single dose, it's various doses. It's a low, a medium, and a high dose. And the trial runs for 6 months to see whether we would reduce proteinuria in an IgA population. And the other trial that's at the moment ongoing that follows a slightly different approach with regard to dose finding is C3G, where we don't test different doses in parallel, but where we have an intra-patient up titration to see whether a certain dose would inhibit the complement system, which would allow you to go directly from this kind of a proof of concept with dose ranging based on biomarkers to go into a phase 3 trial in this rare disease. And with this, I wanted to close and ask whether you have any questions. Thank you very much for your attention.
Bueno, muchas gracias por las presentaciones y vamos a dar comienzo a un par de preguntas y respuestas. A abiertas al público.
Muy bien. Doctor, I would like to start for the first question with Dr. Akelin. As you have nicely shown that TG is an end organ damage, and this in the transplant scenario can come either from T-cell mediated rejection, antibody mediated rejection, or could be relapse or MPGM from another cause. How do you see this is going now? Where are we going now? Are we like designing new trials, targeting treatment to reporting to this findings, or how do you see it from now on?
Of course, the important question is how I'm going to treat this patient. I think the important issue, definitely transplant glomerulopathy is an immune-mediated disease and needs the immunosuppression to be maximized. And what we do is maximize the microvenolar epithelium and keep tacrolimus levels instead of 4 to 7, more 6 to 9 or 6 to 10 range. And if there is significant inflammation, the I score is high, GN and PTC, we give some pulse rate as well. But the important thing is that if there is no antibody, just because transplant glomerulopathy, you do not need to give antibody-mediated treatment to avoid, such as the plasmapheresis, IVIG, rituximab. So in all BAMF meetings, this was my concern, and I said that the BAMF classification is almost like a Bible, and all over the world, they treat the patient. If you say transplant glomerulopathy antibody-mediated, then the patients may receive unnecessary treatment. So this is, I think, the diagnosis is important. Give the adequate immunosuppression, the treatment for T-cell mediated, if it is antibody-mediated rejection, but do not over-treat if this is not the case.
Muchas gracias. ¿Un otro? Doctor Schiavelli.
Gracias por las 3 presentaciones y por el nivel de las 3 conferencias. Yo quería preguntarle al doctor Junge con respecto a la última droga que presentó, la LNP023. Dado que hay alguna experiencia con eculizumab en donantes de órganos para evitar la DGF, si bien no ha habido algunos resultados buenos, han tenido mejor sobrevida, digamos, mejor creatinina de algunos receptores, si usted cree que esta podría ser una indicación de esta droga también.
Yeah, I mean, at the moment I need to admit that our knowledge about the activation of the alternative pathway in transplant is not yet really established. You could speculate that especially in those diseases, for example, if you have C3G leading as a leading cause for transplantation, and you have reactivation of the underlying disease pathology in these patients, then this is most like treatment where you would expect that it helps. And what I haven't shown in the C3G trial that we are conducting at the moment, there are 2 cohorts. One is C3G in native kidneys, and the other one is recurrent C3G in transplanted and immunosuppressed patients. And this is maybe a first cohort where we can generate evidence to not only see whether we can treat the underlying disease or relapsing disease, but maybe have an impact on the outcome of the graft as well. I like the comment, maybe if I could also quickly comment on what you said before, should we treat if we see antibody-mediated rejection? And I think this is one of the underlying ideas for co-stimulatory blockage, because if we have a co-stimulatory blockage in the crosstalk between T and B cells that prevent the formation of germinal centers, and basically B cells can't do the affinity maturation to the epitopes and they can't become a plasmablast, a memory, or a plasma cell, then you basically could avoid any production of antibodies. So I think this is maybe one of the advantages that we will see in future from co-stimulatory blockage trials. And it's interesting to see in the proof of concept, there was not a single patient with the CD40 blockage that developed donor-specific antibodies. That might be an interesting path for future. But thanks for the question.
Doctor del Piano.
Yo le quiero preguntar a la Vale, porque Valeria es un producto de nuestra casa, del hospital privado, y se nos fue y nunca más la tuvimos de vuelta, pero la hemos extrañado mucho, pero me parece que vamos a empezar a trabajar un poco más con ella. Valeria, yo le quería preguntar, referido a lo que comentaba de la situación de epigenética, de la hipermetilación que ocurre en determinado momento de los injertos, y donde parece sobrevenir después una especie de calma en los drivers de regulación de muchas cosas. Esa situación de hipermetilación probablemente usted la ve bien en el laboratorio, pero nosotros en clínica por ahí se nos hace más difícil. Y la primera pregunta que quiero hacer es si se acompaña eso de una sobreregulación de linfocitos T reguladores, que es lo que nosotros vemos en los trasplantes que están en calma. Y la segunda cosa que le quiero decir es que si usted hiciera el experimento reverso, que es darle drogas hipometiladoras, quizás tendría la prueba de concepto con una especie de sobreregulación de todos esos drivers que se duermen un poco con la hipermetilación.
Gracias por la pregunta. Gracias por llamarme la Vale. Creo que no expliqué lo que nosotros evaluamos son los dos caminos. Usamos una red que tiene 1 millón de CpGs diferencialmente expresados. O sea, podemos evaluar 1 millón de CpGs diferencialmente expresados e incluye los dos sentidos, los que están hipermetilados y los que están hipometilados. O sea que podemos verlos instantáneamente los dos. Con respecto a la pregunta de inmunología, nosotros vemos que los marcadores que regulan a los linfocitos T reguladores están hipometilados, indicando que están activados en algunas situaciones que son beneficiosas. Los linfocitos T reguladores siempre han sido, o sea, en los últimos años han sido descritos como los amigos, pero pueden cambiar su rol inmediatamente dependiendo de cuál es el environment en el que se encuentran. No me acuerdo qué otra cosa más preguntó, pero bueno, fuera eso.
Doctor Maldonado.
Sí, una pregunta para Enver. Bueno, hace mucho tiempo que vengo siguiendo los artículos de antibody mediated rejection. Estuviste estudiando el BK por un lado y por otro lado ver la recurrencia de la neuropatía post-trasplante. Ambas cosas se sobreponen en la genómica. ¿Cuál es el camino a seguir? Porque aparentemente hay un límite muy estrecho en donde no se puede diferenciar algunas cosas ya por este método, por el microarrays, en donde se superponen marcadores, transcriptos que no pueden identificar exactamente qué es una cosa o qué es otra. ¿Cuál es el camino a seguir en esto para poder diferenciarlo? También puede ser para Valeria la pregunta.
So thank you for the question. I think the important issue is we do not have the footprints of the recurrent or de novo glomerular disease. I think this is the number one weakest point of the molecular microscope. It is excellent in T-cell antibody-mediated rejection, but as I showed, there is a significant overlap. And even it is available commercially right now, the molecular microscope, but because of that reason, we need to be very careful how to using it. First, there should be greater number of the patients, the centers to find out the specific footprints of the glomerular disease, because even our data is just presented, we have not published it, so there is no published paper to look at the molecular features of the glomerulonephritis. And this is why, at the moment, with the current molecular microscope, I think the only thing is you need to do, whenever you do the biopsy, immunohistopathologically, you will first rule out BK virus nephropathy, easy with SV40 staining, and then to diagnose the glomerular disease, primary, secondary, FSGS, or membranous still, it is very easy, so you do not need the molecular microscope for those reasons. If you exclude these two pathologies, which I believe only makes the 20% of the biopsies, I believe, because the recurrent of glomerular disease 15%, 18%, BK virus 1 to 3%, but the 80% of the time, then the molecular microscope will help. Is it really antibody-mediated rejection or T-cell-mediated rejection? You look at biopsy, non-specific, if a little bit infiltrates there, are you going to increase the immunosuppression, decrease the immunosuppression? Then I think the molecular microscope will help, but first you should exclude the glomerulonephritis.
Voy a expandir un poquito en esa respuesta. Con Enver estamos trabajando juntos en esto y lo que ahora hemos hecho es, estamos haciendo deep sequencing, tratando de ver si hay diferencia más arriba, no en las moléculas efectoras, sino en los upstream regulators. Ayer mostré simplemente una de las libraries, pero lo que hemos hecho con las muestras de Enver es hacer deep sequencing de small no-coding RNAs. Y lo que hemos visto es que son totalmente diferentes a los que regulan la acute rejection o los que regulan los dos tipos, cellular and antibody-mediated. So, posiblemente como biomarker la respuesta va a estar ahí, pero también va a ayudar a discernir los agentes causales.
¿Alguna pregunta más? Si no hay más preguntas, vamos a cerrar la mesa. Agradecer mucho a los speakers por su presencia y sus excelentes charlas y a Novartis por la posibilidad de